So as I've mentioned in previous lecture videos, the synapse is the location where two cells come together to exchange information. Every synapse marks the location where one cell's axon comes within very close proximity of another cell. And every synapse is going to have the same components. So we'll always have a presynaptic neuron that's sending out information. We'll have a synaptic cleft, which is a tiny space between the axon terminal of the presynaptic neuron and the cell membrane of the postsynaptic neuron. And we'll also have the postsynaptic neuron as well. So the presynaptic neuron is the neuron that releases neurotransmitters into the synaptic cleft, and the postsynaptic neuron is the neuron that receives those neurotransmitters and is being acted on by those neurotransmitters. So we can see in this picture on the right, this blue axon is sending signals out to its axon terminals, down its axon, and the synapses are going to be a place where these little axon terminals come within close proximity to our cell, and we're going to release neurotransmitters, and that will then act on this yellow postsynaptic neuron. So in this example, the blue neuron is presynaptic and the yellow neuron is postsynaptic. And in fact, the green neuron and the red neuron are also both going to be presynaptic neurons because they're sending signals out to our yellow neuron. So these are going to be presynaptic neurons because their signal is going to get received by this yellow neuron. Now, like with many other topics, every synapse is going to have both a structural classification and a functional classification. The structural classification depicts which two types of cells are found at the synapse. What is the structure of our synapse? And the functional classification refers to the way that we have information transfer between two different types of cells. So with neurons, these classifications are fairly straightforward. We have the neuromuscular junction, and we learned about this in Chapter 10. This is a synapse between a neuron and a muscle fiber, and we learned about this in great detail in Chapter 10. But we also have neuroglandular synapses, which will be between a neuron and some type of glandular cell. But the most common type of synapse is a synapse between two different neurons. And there are three subtypes of synapses that we will find between two different types of neurons. We have the axodendritic synapse, we have the exosomatic synapse, and the axonic synapse. Again, each of these words are just referring to the two different neurons and which parts of those neurons are forming the synapse. So there will always be an axon that's participating in the synapse. So an axodendritic synapse is between the axon of a presynaptic neuron and the dendrites of the postsynaptic neuron. An axosomatic synapse is going to be between the axon of our presynaptic neuron and the cell body of our postsynaptic neuron. And the axonic synapse is going to be between the axon of our presynaptic neuron and the axon of our postsynaptic neuron. And we actually see all of these different types of synapses shown right here. We can see the axodendritic is between the axon of our presynaptic neuron and the dendrites of our postsynaptic neuron. And we see the axosomatic synapse, which is going to be between the axon of our presynaptic neuron. And you can see that this is now synapsing on the cell body of our neuron. And the axonic is between the axon of the presynaptic neuron and the postsynaptic neuron's axon. So again, don't think too much into these. I know some of these are some big names, axonic and axodendritic, but in the name it's telling you which two parts of our cells are creating the synapse. Now I think axodendritic and axonic are fairly straightforward. However, the axosomatic not so much. So I just want to explain why we call this an exosomatic synapse. Another word for the cell body of our neurons is the soma of our neurons. A soma is the cell body. So an axosomatic synapse is referring to the synapse that involves the cell body of the postsynaptic neuron. So I think the structural classification of our synapses is fairly straightforward. Most of the answers are in the names for these different types of synapses. But we also have a couple different types of functional classifications. And they are the electrical synapse and the chemical synapse. So the major difference between these two types of synapses is how they're propagating their information from the presynaptic neuron to the postsynaptic neuron. So the electrical synapse is a special synapse that has gap junctions, and we see gap junctions in special places throughout the body. We see a lot of these in cardiac muscle. So an electrical synapse is one that has gap junctions. And if you can remember the characteristics of gap junctions, these have tiny little pores that allow for passages of substances between two cells. This is a very, very fast way to transfer signals from one cell to another. So the benefit of having gap junctions is this allows for a group of cells to connect their cytoplasm and to allow for fast signal transmission. And what ultimately comes out of this is the concept of synchronization. We have lots of different cells that are synchronizing their actions together. And that's how our cardiac muscle is contracting in unison all the time. So lots of cells can be coordinated to produce the same function at a given time with gap junctions. And that's what we see at the electrical synapse. Now the chemical synapse is going to be a one-way street. We'll always have information transfer going from a presynaptic neuron to a postsynaptic neuron, and neurotransmitters are going to be what transfers the information from one cell to another. And we actually already learned the chemical synapse in Chapter 10. So we've actually already seen this picture when we were covering excitation-contraction coupling. So we'll just do a quick review of the chemical synapse. When an action potential is fired down the axon of our neuron, that is going to change the membrane potential of our axon and eventually get sensed by voltage-gated calcium channels. When calcium flows into our axon terminals, that's going to signal the release of synaptic vesicles that contain different neurotransmitters. Now in Chapter 10, those neurotransmitters were acetylcholine. However, there's lots and lots of other neurotransmitters that we find in the body. Acetylcholine is the major neurotransmitter used at the neuromuscular junction, but there are going to be some other neurotransmitters that play a role in the synapses between two different neurons. For example, we have dopamine, serotonin, and norepinephrine. Those are 3 examples of different neurotransmitters, and we have some other ones, but those are going to be the major ones that we'll be looking at. So most everything on this slide is something that we've looked at in Chapter 10. We've already actually talked about the chemical synapse when we were going over excitation-contraction coupling. So once those neurotransmitters are released into the synaptic cleft, they're going to interact with ligand-gated ion channels, and that's going to open those channels and allow for influx of different substances in or out of the cell. But the one thing that is new on this slide that I did not cover in Chapter 10 is that chemical synapses can either be inhibitory or excitatory. So this is where we're going to start understanding the concept of graded potentials. So an excitatory synapse is a synapse that transfers an excitatory postsynaptic potential to the postsynaptic cell. Another way we can say this is that an excitatory synapse will generate an EPSP, which is just an excitatory postsynaptic potential. This is some type of membrane potential deviation that will drive our postsynaptic cell's membrane potential towards the threshold potential. So excitatory synapses will always have sodium channels. We'll always see our ligand-gated sodium channels like we saw on this picture. These are ligand-gated sodium channels that allow sodium to flow into the cell down its concentration gradient. So because we have ligand-gated sodium channels, every time our neurotransmitter is released into the synaptic cleft, that's going to cause the opening of these sodium channels. And when sodium channels open, we know that there's a gradient that points towards the inside of the cell. So sodium will always flow towards the inside of the cell, which is a depolarization of our postsynaptic cell. So at excitatory synapses, we're always going to be generating these EPSPs. These are little signals that will slightly depolarize the cell, and when we get enough of these EPSPs that are acting on our postsynaptic cell, that's what's eventually going to bring our postsynaptic cell to its threshold potential. So these EPSPs are graded potential, and they can have different magnitudes. These graded potentials will summate on our postsynaptic cell, and that's what's going to determine whether or not we're going to fire an action potential in the first place. Because every time an EPSP acts on our postsynaptic cell, that's going to slightly depolarize the membrane potential of our postsynaptic cell and make our cell more excitable and ultimately make our cell more likely to fire an action potential. Now we'll also have inhibitory synapses, and these are very, very similar to our excitatory synapses with one major difference. Inhibitory synapses have potassium channels instead of sodium channels. Now we already know that the potassium gradient is pointing from the inside of our cell towards the outside of our cell. So instead of those neurotransmitters coming in, binding to the potassium channels and allowing potassium to flow into the cell, potassium is going to behave along with its gradient. The potassium channels are just going to open up and potassium will flow along its concentration gradient from the inside of the cell towards the outside of the cell. Now when potassium flows out of the cell, that's going to hyperpolarize our cell and make our cell much less likely for firing an action potential. So the signals that are generated at inhibitory synapses are called IPSPs, or inhibitory postsynaptic potentials. These work in the same way that EPSPs work, but these will simply counteract any effect of an EPSP. So if we have an IPSP and an EPSP and both have equal magnitude, that's going to cancel each other out and our cell's not going to change. But let's say we maybe have one or two IPSPs and a whole bunch of EPSPs, that's going to make our cell much more likely of firing an action potential because a lot of excitatory postsynaptic potentials have converged on that postsynaptic cell. So in summary, our excitatory synapses will generate EPSPs that make an action potential much more likely to fire. And inhibitory synapses will generate IPSPs, and that makes our cell much less likely to fire an action potential. So here we will compare the effects of an EPSP and an IPSP, and we'll start with the EPSPs. EPSPs will always cause depolarization, and that is in response to the neurotransmitters that are being released into the synaptic cleft. So when neurotransmitters are released in the synaptic cleft, those neurotransmitters are going to act on sodium channels in the excitatory synapses. Those neurotransmitters will open up the sodium channels, and that allows sodium to flow towards the inside of the cell. Now, our resting membrane potential of a typical neuron is negative 70 millivolts. So when sodium flows in, that's going to decrease the membrane potential, and if we get enough of these EPSPs, that's going to take us to the threshold potential, which is negative 55. And when we've reached that threshold potential, our postsynaptic cell is going to take hold and eventually fire that action potential down its axon. Now at an inhibitory synapse, we're going to have the opposite effect. Inhibitory synapses will generate IPSPs. So when neurotransmitters come into our postsynaptic cell, those are going to interact with potassium ligand-gated channels. And when potassium channels open up, potassium is going to flow out of our cell along its concentration gradient towards the outside of the cell. And this causes a hyperpolarization. We are much less likely to fire an action potential under these circumstances when we're firing IPSPs. So the more IPSPs we have, we're less likely to fire an action potential because that's taking us away from the threshold potential. We would need a lot more EPSPs to counteract the IPSPs that are already present in our postsynaptic cell. So ultimately, what's determining whether or not an action potential is going to fire is the summation of EPSPs and IPSPs. If we can bring our membrane potential to that threshold at negative 55 millivolts, that will allow us to fire an action potential. But if we move away from negative 55 millivolts, that's going to make it much less likely that an action potential is going to fire. So the concept of summation is when we take more than one EPSP or more than one IPSP and we summate their effects. We add up all the effects of the EPSPs and the IPSPs that are all converging on our neuron, and that's what's going to determine whether or not our cell will fire an action potential or not. So there's two major types of summation. We have spatial summation and we have temporal summation. Spatial summation refers to the convergence of different signals based on two different neurons that are in different locations. So you can see that two presynaptic neurons might send two different EPSPs to this postsynaptic neuron. And one of them might not be strong enough to depolarize the membrane potential of our postsynaptic neuron. But if we have a second one, maybe both of those EPSPs are strong enough to fire that action potential. So spatial summation is when two different neurons come together and send their signals at the same time. You can see that in this graph that neuron number 1 might cause a little bit of a depolarization, but it's not going to be enough to fire an action potential because it doesn't get us to the threshold. And then number 2 does the same thing. But when we get more than one neuron generating EPSPs at the same time, that's potentially going to allow us to get our membrane potential above the threshold, and that's going to ultimately fire the action potential. So on the left here we see spatial summation. Temporal summation is a little different. Temporal summation only involves one presynaptic neuron. So temporal summation is where we summate the effects of different EPSPs that occur at different times. So you can see down in this graph We might cause a depolarization with this first EPSP, and then we might cause a second one. But if we fire a bunch of EPSPs, one after another, that might eventually take us to the threshold and allow us to fire an action potential. So spatial summation refers to two different neurons in two different spaces, summating their effects at the same time. While temporal summation just refers to a single neuron that's going to summate its effects over different times. So this is our big picture to sum up postsynaptic potentials and graded potentials. You can see that we have different presynaptic neurons and they're all synapsing on this one postsynaptic neuron. You can also see that neurons 1, 3, and 5 are forming excitatory synapses, which will all contain sodium channels. So those synapses cause a slight depolarization in our postsynaptic membrane potential. And you can see that neuron 2 and neuron 4 are forming inhibitory synapses. So these will contain potassium channels that will make firing an action potential much less likely. So in reality, our postsynaptic neurons are receiving lots of different EPSPs and IPSPs all at the same time. And ultimately, whichever type of signal wins out is what's going to determine whether or not we fire an action potential. And that is going to be decided at the axon hillock, where the axon is arising from our cell body. So let's say maybe we have neuron 1, 3, and 5 all generate their EPSPs. And maybe number 2 generates an IPSP, but number 4 didn't send any signal at all. Well, that's 3 EPSPs and 1 IPSP. One of those is going to cancel out, and that ultimately leaves us a lot more of a depolarization. So that might eventually fire an action potential. But we're always going to need more than one EPSP to generate an action potential. We must summate the effects of these different EPSPs, and that's what will eventually bring us to that threshold potential for our axon to fire an action potential. So I alluded to this concept very briefly in Chapter 10, so we're going to go over this in a little bit more detail on this slide. So it's very important that we have ways that we can remove neurotransmitters from the synaptic cleft of our synapses. If we didn't have ways to remove the neurotransmitters from the synaptic cleft, that would constantly cause our postsynaptic cell to be excited all the time. So it's important that neurotransmitters can be removed from the synaptic cleft so we can turn off any signals that we are sending from the presynaptic cell. So there are 3 different ways by which we can remove neurotransmitters from the synaptic cleft to stop the stimulation of our postsynaptic cell. The first is the simple diffusion away from the synaptic cleft. The synaptic cleft is a space that's continuous with the outside environment of our cells. So those neurotransmitters can simply diffuse away from the synaptic cleft, and they can get cleaned up by other cells. And that's the first way. But the second way is what we saw taking place in our muscles, which was enzymatic degradation. So this is where certain enzymes are going to break down the neurotransmitters that exist within the synaptic cleft. So an example that we have here is acetylcholinesterase, and I briefly mentioned this in Chapter 10. Acetylcholinesterase is an enzyme that breaks down acetylcholine in the synaptic cleft of the neuromuscular junction. It's important that we can get rid of that acetylcholine, otherwise our muscles would constantly be in a state of contraction, and we want to be able to relax our muscles at times as well. So each neurotransmitter is going to have its own enzymes that break it down, and that's our second way by which we can remove neurotransmitters from the synaptic cleft. And the third method by which we can remove neurotransmitters is by uptake or reuptake. Uptake refers to different glial cells taking up the neurotransmitters that have diffused away from the synaptic cleft. But we also have a method known as reuptake, which is where the presynaptic neuron takes up those neurotransmitters and eventually reuses them. So I'd like you to feel comfortable with why we need to have neurotransmitters removed from the synaptic cleft. Again, if we couldn't remove the neurotransmitters from our synaptic cleft, our postsynaptic cell would constantly be active at all times, and we wouldn't be able to turn off that signal going to the postsynaptic cell. So that will take us into different types of neuronal circuits. We're going to briefly go over each of these. And these different circuits are just different arrangements by which certain neurons synapse with each other. So we have diverging circuits, converging circuits, reverberating circuits, and parallel circuits. So we're going to go over each of these on the next few slides. Now there's 4 different circuits we have, but let's not overcomplicate things. Most of these circuits are named based on the types of signals that they are sending. So diverging circuits is where our signal is diverging from one cell to many different cells. we have the amplification of a response due to the increasing number of neurons that are located within this circuit. So we can see that one neuron is going to be synapsing with two other neurons, and this is going to send its action potential down and eventually cause these two cells to fire their action potential. And then those two cells are going to send their signal out to more and more cells, and eventually we're going to have a very, very amplified signal. A great example of a diverging circuit that we've learned is the neuromuscular junction and motor units, when one somatic motor neuron attaches to many different muscle fibers. The signal starts in one cell and diverges out to many different cells. We will also see converging circuits, which is where many different neurons converge on one output neuron. So you can see that we have three different neurons here, and they're all going to synapse with this postsynaptic neuron. So we might see these converging circuits when we're talking about our graded potential. So we might have an inhibitory neuron here, and these two might be excitatory neurons, and they might come in and send their signals, and ultimately our postsynaptic neuron is going to be the one that decides whether or not it's sending that signal out. So the signals in converging circuits are all going to converge on one single postsynaptic neuron. Now we also have reverberating circuits, which are examples of positive feedback loops. So in reverberating circuits, we have neurons that are feeding back on themselves. And that's what continues the signals being sent out. So we'll have one neuron starting off this chain, and this is going to send its action potential down, and that's going to stimulate this one, and then this neuron's going to send its signal down, and so forth. But you can see as we get down here, these neurons are going to send their signals out, and eventually this neuron is going to feed back and potentially increase the signal that's coming from each of these. This neuron is constantly telling these other two neurons to keep firing its signal. So in order for us to turn off a reverberating circuit, we need to interrupt the cycle, and that's why this is a positive feedback mechanism. Our last type of circuit is the parallel circuit. The parallel circuit takes different pathways to get to the same place. So you can see that we start at this neuron, but this neuron might have some axon collaterals here going out to this direction, and then we might have another collateral over here. And so the signal is going to be able to get sent down from this cell all the way to the output cell. But we can also take that signal in a different direction towards the right and left, and we can have different circuits, but they'll all eventually end up in the same place. So because there's different numbers of neurons and different amounts of myelination in each of these different pathways, the signals are eventually going to reach the postsynaptic cell at different times. So parallel circuits will often be used for activities such as mathematics. Now this slide will just serve as a reminder for a concept that I mentioned before in the last lecture video. Neurons have a very, very limited ability to regenerate themselves. If neurons are damaged in some way, it's very unlikely that they will repair themselves back to normal, like much of our epithelial cells do. Our epithelial cells are serving as that covering of our body, and they're constantly at risk for being sloughed off. So we have to be able to regenerate our epithelial tissue. But it's much less likely that we're going to cause some type of damage to our nervous tissue, and that's why our neurons have just lost the ability to regenerate over time. However, while our neuronal characteristic of regenerate ability is very limited, our neurons' characteristic of plasticity is shown very strongly by our neurons. Neurons exhibit this characteristic of plasticity, which means that they are able to produce new projections and form new connections with other neurons. So this characteristic is vital when we want to learn something new. When we learn things, we form new connections and new pathways between neurons. And this is how we can increase our processing speed and learn to become more fluent at certain activities. Now it makes a lot of sense that our neurons exhibit the characteristic of plasticity because these cells need to be able to learn new things and they help us to control our bodies as a whole. But again, our neurons do not have the ability to regenerate if they are damaged. And this is due in part to many different factors. One factor is inhibitory influences that are coming from our neuroglial cells. We also have scar tissue formation that occurs very quickly in the nervous system. So scar tissue will form very, very fast and prevent the repair of these communicative cells. But also, there's a lot of growth-stimulating cues that are no longer present in the adult. While in fetal development, when our neurons are developing, of course we need to have certain cues and growth factors that are stimulating our neurons to grow. However, as we grow, we do not have as many of these stimulating factors. So these are all reasons why neurogenesis does not occur frequently in the central nervous system. Now that being said, there is some indication that some repair does occur to the neurons in the peripheral nervous system. Now this will only occur if the circumstances are appropriate, and there's a few different steps in neuronal repair, but the major step I want you to focus on is Wallerian degeneration. As long as the cell body of our neuron is intact and the axon is what is damaged, we can actually degenerate the latter part of our axon and form a regeneration tube with Schwann cells. And we can eventually repair the axon of the neuron that is existing in the peripheral nervous system. However, again, I will mention that this only occurs if the circumstances are appropriate, if our Schwann cells are functional and we haven't formed too much scar tissue quite yet. So while there is some capability of repair in the peripheral nervous system, nervous tissue as a whole does not really exhibit regeneratability very strongly. Now these are just a few clinical terms that I'd like to introduce you to, since many of you are going on to nursing school and medical school. So a neuroblastoma is a tumor that occurs in nervous tissue. This suffix "oma" refers to a tumor, and we'll often see this when we talk about cancer. Blastoma is some type of tumor, so a neuroblastoma is a tumor of nervous tissue. And we also have neurotoxins, which are going to be substances that are poisonous to the nervous system. So I just post these terms so that you can take a look at these and familiarize yourself with some of the terms that you'll likely be seeing in your future careers. Now there's a few different disorders that we're going to discuss to finish up Chapter 12. And the disorders that we're going to discuss are multiple sclerosis, epilepsy, exocytotoxicity, and depression. All of these conditions in some way affect the nervous system. So we'll start with multiple sclerosis. Multiple sclerosis is a condition where the myelin sheaths of our neurons are breaking down. And these myelin sheaths specifically occur at the neuromuscular junction. So if our myelin sheaths are breaking down, that's going to significantly slow the transmission of our action potential to our muscle fibers. And we call this condition multiple sclerosis because under the microscope you can actually see the scleroses that are present in our nervous tissue. So this is actually an autoimmune disease where our own immune system is attacking the Schwann cells that make up that myelin sheath. Now there's no cure for this condition, unfortunately, but we do have certain treatments. And one of the treatments that I'd like to share with you is beta interferon. As I mentioned, beta interferon is not a cure for this condition, however it does slow the progression of the disease. Exocytotoxicity is our next condition. This condition refers to the degradation of neurons due to prolonged activation of our excitatory synapses. We have too much excitatory stimulation caused by increased levels of the amino acid glutamate. Now you don't need to know all the ins and outs of this condition, but I do want you to be aware that high levels of glutamate are caused by oxygen deprivation to the central nervous system. And ultimately, what most often causes oxygen deprivation is a stroke. Some type of blood clot in the brain causes an inadequate blood flow in a certain area, and that can potentially cause exocytotoxicity. Now, another condition that affects the nervous system is depression. Depression is characterized by feelings of sadness and helplessness, as well as suicidal thoughts. With depression, there is decreased neurotransmission taking place, so the central nervous system is not as active as it should be. So this will ultimately depress the central nervous system and depress the individual as well. So one of our first-line treatments for depression are a class of drugs known as SSRIs, which stands for Selective Serotonin Reuptake Inhibitors. Serotonin is another neurotransmitter that we have in our nervous system, and this is the neurotransmitter that makes us feel happy. So oftentimes in depression, there's not enough serotonin being released at the synaptic cleft. So by using an SSRI, we're able to prolong the activity of serotonin in the synaptic cleft. Essentially, we're blocking the reuptake of serotonin, so serotonin exists within the synaptic cleft for a much longer time. Now, there are other classes of drugs that can be used to treat depression as well, but this is one of our first-line treatments. Now, lastly, for our conditions in Chapter 12 are two viral infections. These two viral infections are rabies and shingles. Now, I'm going to leave these two conditions for you all to self-study, and to look at in your books. And that will conclude our lecture video on Chapter 12. We will come back for our next video, which will cover Chapter 13 and the spinal cord.